Assalamualaikum and greetings. My name is Fathin Afiqah Binti Zainuddin. I'm going to present my final year project entitled A Review, Magnetic Solid Phase Extraction and Dispersive Solid Phase Extraction of Fluoroquinolones Antibiotics in Biological and Food Sample. My supervisor is Madam Dyia Syaleyana Binti Shukri. Background study. Antibiotics are antimicrobial substances with antibacterial and antifungal properties that are used to treat infections in both people and animals, as well as protozoan infections in some instances. It was discovered in 1928 and widely regarded as the most important medical development of the 20th century. Quinolones are a class of antibiotics with a bicyclic core structure that is related to 4-quinolone. Nalidixic acid was the first quinolone antibiotic to be discovered, and it was first published in 1962 to treat both community-acquired and severe hospital-acquired infections. Problem statement. Due to excessive use of quinolones class in veterinary and human medicine, particularly in animal production units, a global environmental pollution problem has arisen known as antibiotic contamination. According to the Food and Drugs Administration (FDA), excessive use of fluoroquinolones will associate with disabling and potentially permanent serious side effects on the tendons, muscles, joints, nerves, and central nervous system of the body. Fluoroquinolones residue that are coming from aquatic farm, hospital, and sewage effluents, industrial wastewaters, and untreated wastewater are eventually migrated and reacted in the aquatic environments, which in turn can harm mankind and environment. Significant study. Excessive use of fluoroquinolones may cause serious adverse effects towards humankind and environmental, which perhaps resulted permanent negative effects on the body's tendon, muscles, joints, nerves, and control nervous system. This review highlighted the application of magnetic solid phase extraction (MSPE) and dispersive solid phase extraction (DSPE), which gained a lot of interest from researchers as one of the best methods to extract fluoroquinolones from biological and food samples. These methods are easy to implement, low cost, and provide high extraction efficiency with low limit of detection (LOD). There are two objectives of this review. First, to highlight the potential application and adverse effect of fluoroquinolones towards mankind. Second, to critically discuss the application of magnetic solid phase extraction (MSPE) and dispersive solid phase extraction (DSPE) techniques for the determination of fluoroquinolones in biological and food samples. Literature review. Fluoroquinolones are the derivative of quinolone, which are fluorinated at C6 position of the quinolone ring. It is a type of antibiotic that have been widely utilized for the treatment or prevention of certain bacterial infections in human, livestock, and aquaculture industries. The usage of fluoroquinolones compound in animal husbandry result in the production of antimicrobial resistant bacteria. According to multiple studies, majority of antibiotic resistant E. coli identified in human originated in food. Animals, specifically chicken. Antibiotics were graded by the World Health Organization based on their usefulness in human medicine, with fluoroquinolones regarded critically important to lessen human health risks connected with their usage in veterinary applications. Adverse effect of fluoroquinolones. Fluoroquinolones have an excellent safety record, with most side effects being minor. A safety review conducted by the Food and Drugs Administration (FDA) found that fluoroquinolones were associated with disabling and potentially severe harmful effects on a permanent basis. The adverse effects are detrimental to tendons, muscles, joints, nerves, and human's body central nervous system. The skin, liver, and nervous system are the most affected systems by fluoroquinolones toxicity. The most well-known adverse reaction is phototoxicity, the risk of which varies greatly between fluoroquinolones, lomofloxacin, and sparfloxacin, which are particularly dangerous. Phototoxicity is caused by a chemical reaction between light and drugs. Nitrotoxicity occurs as well with a wide range of incidence among the various compounds. Serious side effects of these drugs have been rare but have included interstitial nephritis and grand mal convulsion. Safarikova and Safarik developed the first analytical application and the first use of the term MSPE for the separation of copper tyrosine dibisylnitrate magnetite in 1999, which was published in the Journal of Analytical Chemistry. When compared to SPE, it is significantly less complicated because before analysis, the analyte must not be stored in column and cartridge. For the extraction of materials, only a powerful external magnetic field is employed. Using a solution or suspension containing the analytes of interest in MSPE, selective adsorption on the solid surface is achieved through the analyte's direct contact with the magnetic solid phase nanoparticles, MNP, incorporated in the solution or suspension. Iron oxide is commonly used MNP in removing organic contaminants due to its high surface area, small particle size, good paramagnetic characteristic, dispersivity of water, stability, and low toxicity, as well as it is of its synthesis and functionalization. The MNP must be modified in addition to enhance its effectiveness of adsorption and realize its full potential in the remediation of fluoroquinolones contaminated in food and biological samples.
Table 3 and 4 summarize magnetic solid phase extraction of fluoroquinolones in biological and food sample. Table 3 shows in 2021 and Zainuddin et al. Used MSPE coupled with multivariate calibration methods to determine the presence of fluoroquinolones in the plasma, hospital waste, and fever water. And Lee et al. In a year of 2018 carried out a research to remove organic pollutants from biological samples. In their research, graphene-based magnetic composites were used as MSPE adsorbents. The range of LOD for this paper were 0.05 to 0.3 nanogram per gram, and the percentage recoveries were in the range 82.4 to 108.
When Chile and Dyia et al. Had developed a sulfonate functionalized covalent organic framework-based magnetic sorbent for determination of fluoroquinolones in pork, chicken, and bovine. For fluoroquinolones analysis, the new MSPE approach in conjunction with HPLC-MS achieved low limits of detection, which is 0.1 to 1 gram per kilogram when used in the most favorable conditions. Furthermore, the disclosed approach was successfully employed to remove fluoroquinolones from contaminated food, which is pork, chicken, and bovine, with a successful recoveries around 82 to 110%. Dispersive solid phase extraction (DSPE) has become a widely accepted technique because of its widespread application since its inception around the year 2000 for extraction, isolation, and cleaning in the analytic treatments of wide range of drugs used in industry of livestock. DSPE simplifies SPE cleanup, allowing for the detection of more samples at the same time, extremely fast, and requires less solvent consumption. In DSPE, a solid sorbent, typically silica or polymer base, is directly added and dispersed into the sample solution, which increasing the surface area of sorbent and target analyte through dispersion process. A variety of chemical compounds have been added to the sorbent used. in the SPE to change their affinities for antibiotics residues, resulting in solids that have been chemically modified. All of these adjustments boost analyte selectivity to ensure maximizing retention while avoiding interference with the analytical matrix. Table 5 shows dispersive solid phase extraction of fluoroquinolones in biological samples. In 2021, R1 et al. Reported a highly selective extraction using microdispersive DSPE method combined with an improved sensitivity detection by quick-column diffusion injection and liquid chromatography high-resolution mass spectrometry for determination of fluoroquinolones in swine body fluids. The proposed method demonstrated good linear coefficients greater than 0.99 as well as high sensitivity with the FOD. The concentration were in the range of 0.02 to 0.03 microgram per liter with percentage recovery 85 to 100 × Conclusion and recommendation. The SPE and MSPE were used for extracting the fluoroquinolones in biological and food samples. Fluoroquinolones antibiotics are widely used in livestock industry, and excessive use of it can cause adverse effects to environmental and humankind. The solvent used in MSPE also being utilized using various materials in order to increase its analytical performance. The ability of the solvent to extract fluoroquinolones from different samples was compared. MSPE considered as the effective technique in removing fluoroquinolones due to its low cost and low solvent use. Most of the solvent used in MSPE also considered environmentally friendly. The range percentage recovery of solvent were 82.4 to 108.5. From the percentage recoveries, it shows that the fluoroquinolones were successfully extracted using the MSPE and DSPE. It is important to make sure that the fluoroquinolones level in environmental and food at the safe level to avoid any adverse effect on human, animals, and environment. Since the excessive use of fluoroquinolones in industry increasing nowadays, the further study need to be conducted to detect more side effects and find the most efficient solvent to extract fluoroquinolones in a simple and fastest way.